So, good morning, good afternoon, or good evening, or even a very early morning, depending on where in the world you are. I'm Max Häggblom. I'm the editor-in-chief of FEMS Microbiology Ecology, and it's my distinct pleasure to welcome you to this webinar on the sponge microbiome. Never underestimate the power of the microbe, to paraphrase Louis Pasteur. With the global coronavirus pandemic making traditional conference attendance impossible, FEMS is launching today a series of webinars to support the microbiology community during this time. These webinars will provide a forum for the presentation and discussion of key research, enabling the flow of ideas to continue despite the cancellation of so many in-person events. Today's webinar launches the series that will continue later with polar and alpine microbiology on July 23rd and environmental dimension of antibiotic resistance on August 6th. These webinars are also linked to thematic issues on these topics in FEMS Microbiology Ecology, so there's much more to explore in the journal. Today, we focus on the sponge microbiome. Sponges are the oldest extant multicellular animals. They're more than 600 million years old, and they are now well recognized as holobionts, comprised of the host and its symbiotic microbes. Marine sponges are ecologically important components of coral reef ecosystems, where they provide habitat for a wide range of species and couple the benthic and pelagic zones through their seawater filtration capability. Today, we will hear about the importance of microbially mediated nutrient cycles in marine sponges, about some of the distinct adaptive features in symbionts, their genomes, and the establishment of a sponge symbiosis model systems. We have three excellent speakers and a global audience, so I'm very excited about this webinar. Our first speaker will be Russell Hill from the Institute of Marine and Environmental Technology, University of Maryland Center for Environmental Science, followed by Laura Steindler at the Leon Charney School of Marine Sciences at the University of Haifa, and then Rodrigo Costa from the Department of Bioengineering of Instituto Superior Técnico at Universidad de Lisboa. After the talks, we will then open the session for questions and discussion. So you can use the chat function to send in questions, and we'll continue with discussion for all of the presentations. So to begin, it's my pleasure to introduce Russell Hill, a professor at the Institute of Marine and Environmental Technology at the University of Maryland Center for Environmental Science in Baltimore, Maryland in the USA, and he will talk about microbially mediated nutrient cycles in marine sponges. Russell.
Thanks very much, Max. I thank you and Sarah and everyone at FEMS Microbiology Ecology for putting together this great webinar. And I want to give you a quick overview of what I plan to talk about. I'm going to do a short introduction on sponges. I'll then talk about some of our early work on nitrogen cycling, in particular nitrogen fixation by the bacterial symbionts of sponges. A little bit more detail on more recent work on the phosphorus cycle, and a brief summary. And with that, let's go straight into a quick overview of sponges. So this image is taken at a coral reef in the United States, in Florida, at Key Largo. And as is the case with many coral reefs, you see some coral, but also you see a lot of sponges. This magnificent barrel sponge in the foreground, Zestospongia muta. Here's a smaller sponge, Mycale laxissima, and other sponges scattered around here. And these are really prolific organisms found in oceans around the world that are now over 6,000 formally described species. Most of them are marine, but there are around 500 freshwater sponges. They have a cellular level of organization. And I'll illustrate that. If you take a typical sponge, this is a sponge about the size of a football. It's a sponge called Cinia strobilina. And if we think about that in terms of its cross-section, the sponge has a number of small holes on the surface of the sponge, and it takes in the surrounding seawater. These specialized cells called choanocytes make the sponge a very efficient pump. The water passes through the sponge and then out the top of the sponge. And during that process, the sponge has access to all the bacteria in the surrounding seawater, as well as the viruses and a lot of dissolved organic material. And this is where the sponge is getting its food from. So this water, the surrounding water, passes through the sponge, it's through being pumped by these choanocytes, and then goes out the top of the sponge. And the water emerging from the sponge has a much, much lower bacterial content than the water going in. So another point that I want to make about sponges, this is one of, I think, the first sponge I ever worked on, and this was quite a few years back together with my graduate students at that time, Nicole Webster, who has gone on to do so much wonderful work on sponges and has been a huge contributor to the field. This sponge is called Rhopaloides odorable. It's from the Great Barrier Reef in Australia. And we studied the microbial community in the sponge in some detail. And to begin with this TEM, you see that the sponge is packed full of microbial cells. Most of these are bacterial cells, and you immediately get an idea of the huge diversity of microbes associated with the sponge. Now, at this point, we know a huge amount about the microbial diversity associated with many hundreds of sponges from all around the planet. And this is just one example. Nicole Webster was the last author on this paper. The first author is Thorsten Thomas at the University of New South Wales. They studied the microbial communities in many different sponges as part of a big consortium. And a few points. Some microbes appear in many different sponges. You'll see these proteobacteria up near the top, the gamma proteobacteria and the alpha proteobacteria that we're going to hear a lot more about from Rodrigo. Also, this does include actinomycetes, but also archaea. So most sponges do contain archaea as well as bacteria. So that's one point, a great diversity of microbes. The second point is that each sponge has its own characteristic microbial community. And these communities tend to be very stable in space and time. So if you go back and sample the same sponge from different regions or different times of the year, you'll find that the microbial community is remarkably consistent in many sponges. One more point that I want to make about sponges is that they are very efficient pumps. I already touched on that. There's a long history of studying water pumping by sponges, and many famous sponge microbiologists have studied this, notably Jean Vasselay early on. In this image, this diver has squirted a small amount of a harmless fluorescent vegetable dye at the base of the sponge. And within a few seconds, the sponge has transported that water through the sponge and is rapidly pumping water out the top. So 1 kilogram sponge can pump somewhere between 10,000 and up to 40,000 liters of water every 24 hours. This means that sponges are also great couplers between the benthic and the pelagic environment. So they are potentially taking material from the benthos and pushing it out into the water column. And to move towards some talking about nutrient cycling, I want to highlight this very important work by Jasper de Goy and colleagues. This was published in Science a few years back. Of course, this is building on a long history of studying the role of sponges in nutrient cycling, in particular their feeding on dissolved organic matter. And some of this work was done by a famous sponge biologist, Henry Reiswig, who very sadly died just a few days ago, and he's a big loss to the sponge community. In this great paper by de Goy and colleagues, they showed that sponges in coral reef environments have a pivotal role in taking dissolved organic carbon into a loop that they call the sponge loop, and effectively concentrating this dissolved organic carbon and making it available to other organisms in the coral reef environment. So that's a key point. That's one I'll come back to. And I recommend to you a very recent paper just published in ISME-J. Laura Ricks is the first author. Jasper de Goy is an author on this as well, as is Uta Henschel, who's done so much work on sponges. And in this paper, they show very convincingly in an elegant study that dissolved organic matter is the main source of carbon for many sponges, and it can be up to 90% of the carbon in the diets, the heterotrophic diets of sponges. And it's taken up by the bacterial symbionts and also by the choanocytes, the cells that I told you about that pump so efficiently. So that's a little bit of background on carbon cycling by sponges, their role in cycling carbon in the coral reef environment. I want to talk now briefly about nitrogen cycling. And I'm going to focus on the process of nitrogen fixation. So sponges take in inorganic nitrogen in the surrounding water. And because of their bacterial symbionts, these symbionts have the ability to fix that nitrogen using the key enzyme, and NIFH, the nitrogenase enzyme, and produce ammonia that can be used by the sponge or potentially can be available to other organisms in the coral reef environment. So this is a really important process in the nitrogen cycle. Sponges have been shown to harbor symbionts and to be involved in many other steps in nitrogen cycling as well. But to focus just on this one process, we studied this quite a few years back in 2 sponges from Florida, Mycale laxissima and Yersinia strobilina. And one of the things we did was to study the delta N15 values. This value gives you an idea of how much of the nitrogen in the sponge comes from biological nitrogen fixation. And in the case of one of our 2 sponges, Mycale laxissima, a fair amount of the nitrogen was from biological nitrogen fixation. The second sponge, Yersinia strobilina, was getting almost all of its nitrogen through this process of biological nitrogen fixation. So we studied that further, and my ex-PhD student, Nagla Mohammed, was the lead person in these studies. And she did something that I believe was a first. She was successful in isolating messenger RNA from the sponge microbial community and reverse transcribing that. And this is specifically the messenger RNA that encodes the NIFHG. I believe this is the first time that the expression of any bacterial gene was shown through this process in sponges. If anyone wants to contradict me on that, please do that during the question time. But this was a very exciting result for us. Once we had the cDNA, we could clone that and we could get an idea of which bacteria were important in the nitrogen fixing process. And we found that around 50% of the NIFH genes being expressed in both of these sponges were closely related to NIFH from cyanobacteria. And alpha proteobacteria also play a big role. So cyanobacteria were key. We went on and Fan Zhang, who worked on this project for me during his PhD, as well as Jan Vicente, did a really elegant study over 24 hours looking at changing patterns of NIFH gene expression during a 24-hour cycle. And it was remarkable how dynamic that was, with cyanobacterial NIFH dominating at some times in the day, and proteobacterial NIFH gene expression being highly dominant at other times in the day. So I think this is a really interesting thing to be thinking about. I told you that the bacterial communities tend to be very stable. However, patterns of gene expression are very dynamic and need to be studied on a temporal cycle with very short time periods between sampling. So some key findings on our work on nitrogen fixation. Cyanobacteria were very important in this process. There were dynamic changes in the diazotrophic community. We were also successful in actually growing many of the nitrogen fixing bacteria, including a fascinating filamentous cyanobacteria that took us around 2 years before we were able to successfully isolate that and get that into pure culture. And that filamentous cyanobacterium turns out to be important in some of the work that I'm going to tell you about shortly. So now I want to move on to our work on the phosphorus cycle in marine sponges. And this was exciting because it came about completely by accident. My student Fan Zhang, who I already told you about, was, for reasons we still don't understand, he was really battling to visualize the bacteria in sections of a barrel sponge, of Zester Spongiomuta. So he did something unusual without telling me. He tried 10 times the standard concentration of DAPI, the stain that we often use to visualize the bacteria, and also 100 times the standard concentration of DAPI. And he looked at those sections, and for the first time ever, he saw a whole lot of yellow fluorescence. We'd never seen this before. Unfortunately, he didn't ignore that. A fan and a postdoc in my lab at the time, Leah Blasiak, dug into this a little bit, discovered that DAPI binds to DNA and fluoresces blue, as we all know, but also at high concentrations of DAPI, it can bind to this compound called polyphosphate. And under those conditions, it will fluoresce yellow. So this was an intriguing hint that we may be seeing polyphosphate in marine sponges. Polyphosphate is a long chain molecule. N can be 60 or 80 or even 200 units. And it's a molecule that stores phosphorus and also stores energy. So as polyphosphate is broken down into individual units, ATP is formed. So you can think of this as a cellular battery, a very efficient way of storing energy as well as phosphorus. So we went on to investigate that further, and we used a technique called SEM, energy dispersive spectroscopy, to investigate what some of those yellow particles were. This particle here, labeled A, you can see is rich in phosphorus. This particle is overlaid on a specule, and you see the specule clearly here. In that case, we also saw phosphorus. We saw silica, as you would expect from a glass specule, and we saw some calcium. We used various other approaches to confirm that sponges can be rich in polyphosphate. In fact, between 25 and 40% of all the phosphorus present in sponges, so that's in all the sponge cells, their membranes, their DNA, and all the bacterial symbionts, their DNA and their membranes, 25 to 40% of the phosphorus can be present in the form of the storage compound, polyphosphate. We separated out the symbionts from some of these sponges, and you can see the polyphosphate granules within the sponge symbionts. So the symbionts are key in producing the polyphosphate. We came up with this model. We already talked about the water being pumped through sponges, and the dilute inorganic phosphate, or possibly the phosphate present within the bacterial diet, is taken up by bacterial symbionts, and under aerobic conditions, polyphosphate can accumulate. And we hypothesize under anaerobic conditions that polyphosphate can be broken down to release energy as well as inorganic phosphate. There are a number of possibilities, number of fates for this phosphate. One possibility is that this combines with calcium to form apatite, which actually takes this out of the biological cycle. It makes it insoluble mineral. So we're really interested in this process of phosphorus cycling in sponges. Briefly, so far, we've showed that phosphorus sequestration in the form of polyphosphate is mediated by sponge symbionts. Sponges may play a role in concentrating the very dilute phosphorus in the surrounding water and potentially make it available to other organisms in the coral reef in the same way as Jasper de Goey and colleagues showed for concentration of carbon by sponges. And sponges are an intriguing new source of interesting bacteria that are able to accumulate polyphosphate. And we went on and wrote a review on the topic of nutrient cycling by marine sponges, considering carbon, nitrogen, phosphorus, and sulfur. And the co-first authors on this review are Fan Zhang and my current graduate student, Lauren Jonas, who is continuing to study phosphorus in sponges. I'm going to tell you a little bit about her work, but briefly, I want to go into the slightly more complicated model. We already talked about parts of this, where bacterial symbionts can take inorganic phosphate under aerobic conditions and concentrate it in the form of polyphosphate, an energy and phosphorus storage compound. We hypothesize that under anaerobic conditions, this polyphosphate can serve as a source of energy for the highly energy requiring process of nitrogen fixation. So this is an intriguing possibility to investigate further. We know that nitrogen fixation occurs under anaerobic conditions. Also, one symbiont that we've succeeded in cultivating, this filamentous cyanobacterium that I mentioned, has the ability to fix nitrogen. That same filamentous cyanobacterium has abundant polyphosphate. So it's tempting to hypothesize that that's a key player, both in phosphorus storage and in nitrogen fixation. And we are looking at that further now. We are looking at this process in the lab, studying rates of uptake of inorganic phosphate, dissolved organic phosphate, and also phosphorus provided in the form of bacteria in the sponge's diets. We're using this using radio-labeled phosphorus P32. So we've set up tanks in the lab. You'll see in this experimental setup, our sponges are in this inner tank. The outer tank is a safety precaution in case of any leaks because of the radioisotope that we're working with. And these experiments, I think, have been going very well, a lot of fun. And Lauren Jonas, I mentioned, my current graduate student, is actively involved in these, and I'm going to share just a little bit of her preliminary data from the work that she's doing. And in this preliminary study on 4 individual sponges, looking at rates of uptake of organic phosphorus in the form of ATP that we inject into the water, and also inorganic phosphate, we see uptake of phosphorus in both of these forms fairly rapidly over a period of 6 hours, and this uptake does continue. In fact, inorganic phosphate continues steadily, and the rate of uptake of the organic phosphorus levels out to some extent. So those are ongoing experiments generating a lot of exciting data. And briefly to conclude, I want to come back to this role of sponges as important nutrient cyclers in the marine environment. And actually, this goes all the way back to a question posed by Charles Darwin. He was struck when he looked at coral reefs about this issue of how such high production can be present in an ecosystem where the surrounding water is so very low in nutrients. And this has been stated as Darwin's paradox. We believe that there is data emerging on carbon, including the work that I told you from Jasper de Goey and colleagues, on nitrogen from our lab and many other labs, and now more recently on phosphorus, that sponges can concentrate these important elements from the very dilute surrounding water, and they can effectively be packaging these elements and making them available to other organisms in the coral reef environment. And I'll end there by thanking some of the many people who've worked on sponges with me, starting with Nicole Webster, who's gone on to do such great things. And the work that I talked about today was Nagla Mohammed on the nitrogen fixation, Fan Zhang, Lauren Jonas, and Jan Vicente on some of the work on nitrogen and also phosphorus. And I thank you for your attention. And with that, I'll hand back to Max.
Thank you, Russell. This was a great start to the webinar, and thank you for really giving a wonderful overview of the nutrient cycles, and with a focus on nitrogen, phosphorus, as well as carbon. Now from here, we're going to go to the University of Haifa in Israel, where Laura Steindler will present her work on genomics of sponge symbionts, and also how they're working on establishing a sponge symbiosis model system. So Laura, I think you're all set up to take charge.
Thank you very much, Max, for organizing this webinar and for inviting me to take part into this, first of all. And I will talk indeed today about genomics of sponge symbionts and trying to establish a model system to study sponge symbiosis. So, as Russell just mentioned, the sponges are inhabited by a very, very diverse community of microorganisms. And in these STEM images, you can see this diversity. And what is interesting to us is what enables them to be symbionts to the sponge, what makes them interact in a positive way with the sponge host. And some of the questions that we ask is how do microbial symbionts escape the phagocytic cells of sponges? Russell mentioned that sponges filter incredible amount of seawater, trapping bacteria and phagocytizing them as a source of food. Yet in the same sponge matrix, we have sponge symbionts that are not digested by the host sponge. So to try and answer these and similar questions, we use different approaches. One is to look at the genomic features of symbionts and see how they differ from genomic features of free-living bacteria that are food to the sponge. And another approach is to look at it from the host side, and there to try and establish a system where the presence and absence and the acquisition and loss of symbionts can be controlled. Early studies in sponge symbiont genomics started about a decade ago and utilized comparative metagenomics, meaning that the DNA sequence from all the microbial community inside the sponge was compared to the DNA extracted from the whole microbial community from seawater collected near the sponge. And this gave an average picture of the genetic features that are either enriched or depleted in sponge symbionts. Among the interesting insights from these early studies was an enrichment in eukaryotic-like proteins, which is a class of proteins that are found in prokaryotes. However, from an evolutionary point of view, they likely have an origin in eukaryotes. And both in sponges and in other symbiotic systems, they are postulated to mediate host microbiome interactions. Later studies, thanks to the development of bioinformatic pipelines, enabled to investigate the genomes. So specifically to assemble genomes of single symbionts inside the sponge host. And today I will focus on this guy here. And even in transmission microscopy images, we can recognize those as they are cyanobacteria. And they have this typical circular thylakoid membranes where photosynthesis occurs. And Russell talked earlier about filamentous cyanobacteria that can fix nitrogen. Well, I will talk about unicellular cyanobacteria that do not have the ability of fixing nitrogen. And the dominant Synechococcus species in sponges worldwide is called Synechococcus spongarium. Spongarium is widespread both in terms of geography and in terms of sponge species. So in the graph to the left, you see 42 different sponge species collected all over the world that were shown to harbor Spongarium. Differently, a closely related symbiont, a closely related Synechococcus symbiont of sponges is Synechococcus feldmani. And this symbiont is unique to one sponge species called Petosia ficiformis that grows both in the Mediterranean Sea and in the Eastern Atlantic Ocean. Additional differences between these two types of Synechococcus symbionts include their type of transmission. So Spongarium is vertically acquired, meaning that it is transferred to the next sponge generation through sponge gametes, sponge eggs, sponge sperm, and it can be detected in the larval phase of the sponge. Differently, the larvae of Petosia ficiformis are devoid of any type of symbiont, and it is considered that this sponge acquires all its symbionts from the environment. Furthermore, Spongarium is extracellular and feldmani is intracellular, and it is found together with a diverse microbial heterotrophic community in specialized cells called bacteriocytes. An additional interesting feature of Petosia ficiformis is its facultative nature with the symbiont feldmani. So when the sponge grows in light environment, it will appear pink thanks to the presence of the Synechococcus feldmani, while when it grows in underwater dark caves, it will appear white due to the lack of the photosymbiont. So we sequenced the genomes of feldmani, Spongarium, and we compare them to those of free-living cyanobacteria. And this was in order to look at which genetic features enables these different lifestyles that I just described. Doing a phylogenomic analysis, we found that feldmani and Spongarium share a common ancestor from which they evolved and diverged through the different lifestyles that I just presented. And together, they are sister to the free-living marine clade Proclodococcus synechococcus. As of now, I will use a purple symbol to refer to the environmentally acquired feldmani, a green symbol to refer to the vertically transmitted Spongarium, and a blue symbol to refer to the free-living cyanobacteria. So when we look at the shared genetic features between feldmani and Spongarium, we can get an insight of what are the genetic features that enable life inside and in interaction with the host sponge. Among these features, we found eukaryotic-like proteins. However, the type of eukaryotic-like proteins in the feldmani and in the Spongarium were different, with feldmani being enriched in fibronectin type 3 domains and in cadherin domains. And both these two types of domains were shown to have cell adhesion properties. For example, bacterial pathogens use fibronectin type domain proteins to attach to epithelial host cells. And accordingly, we suggest that these two types of domains might enable feldmani to colonize Petosia ficiformis and possibly to be internalized in the specialized bacteriocyte cells. On the other hand, we know that feldmani has a dual lifestyle being acquired from the environment, and thus genetic features that are shared between feldmani and the free-living cyanobacteria can tell us something on what enables feldmani to survive at times it is outside its own host. Among these are genes related to response to environmental stress, where we believe that inside the sponge, the temperature and the light are more stable, and the feldmani will need to also be able to cope with a difference in condition when found outside the sponge. Furthermore, we found that feldmani and free-living cyanobacteria share cell surface features, and they differ from those of Spongarium. In fact, Spongarium was found to lack 4 genes in the biosynthetic pathway of the sugar DTDP-L-rhamnose. And DTDP-L-rhamnose happens to be the sugar in the O antigen of which is the outmost part of the lipopolysaccharide found on the outer membrane of cyanobacteria. Now, the fact that the symbiotic Spongarium does not have the ability to produce rhamnose suggests that the O antigen of the symbiont differs from the O antigens of free-living cyanobacteria. So we looked at the literature to understand what is the function of the O antigen in free-living cyanobacteria. And we found that in freshwater cyanobacteria, the O antigen is involved in predator-prey interaction, where a mutant of the freshwater cyanobacterium in the O antigen becomes resistant to grazing by amoeba. Furthermore, the same mutant that becomes resistant also loses its capacity to float in seawater, in freshwater, and sinks to the bottom of the tube. The sponge predator cells that digest and phagocytize bacteria are also called amebocytes, as they are functionally similar to amoeba. And all these brought us to hypothesize that the O antigen, including the L-rhamnose sugar, might be recognized as a food receptor by the sponge cells, and therefore that the lack of O antigen in symbiotic cyanobacteria, in Spongarium, may enable it to survive sponge digestion. Unfortunately, unlike the filamentous cyanobacteria that Russell described, nobody has been yet able to grow in culture cyanobacterial symbiont, unicellular cyanobacterial symbionts. And therefore, what we try to do to investigate these genomic features is to create mutants in freshwater cyanobacteria that will mimic the specific symbiotic features. In this case, we have grown Synechococcus WH8102, a free-living cyanobacterium. We knocked out, disrupting the gene involved in rhamnose production, and we grew the mutant. This is how the wild type grows, while the mutant will lose its floating ability and sink and grow at the bottom of the bottle, reminding us of what we have seen in the literature about the freshwater cyanobacterium. Together, we came up with this model, suggesting that Spongarium is transferred to the next sponge generation through the larvae of the sponge, and therefore does not require floating capacity. And the lack of an O antigen with rhamnose will enable it to be protected from amebocyte phagocytosis. Differently, Feldmani is supposed to be acquired from the environment and does require the typical O antigen with rhamnose. And then, when it is found inside the sponge, in order to avoid phagocytosis by its own host, it will need to be internalized in the specialized bacteriocyte cells. However, so far we have not detected sequences of Feldmani in seawater, nor have previous studies been able to show acquisition of Feldmani by white Petlosia fisciformis sponges. Rather, when Petlosia is transferred from light to dark environment underwater, it will lose the cyanobacterial Feldmani symbionts, yet it will survive, differently from the sponge Chondrilla nucula, which hosts Spongarium and that shares the same habitat as Petrosia, but when transferred to a dark cave, it will lose Spongarium and it will die. This suggesting a different type of interaction, facultative for Feldmani and obligatory for Spongarium. We tried once again to see whether we could obtain the acquisition of the highly specific Feldmani symbiont by Petlosia, this time transplanting white sponges of Petlosia to dim light rather than to highlight environments. After 4 months, we saw nothing, but after 8 months, we were very excited for the first time to report the acquisition of the highly specific symbiont by observing these first pink patches. And later months, the sponges were fully colonized by the highly specific symbiont. The symbiont is a photosynthetic symbiont, so it produces organic carbon, which might be utilized by the heterotrophic microbial community that shares the bacteriocyte. We thus imagine that it might affect the microbial community, and we looked whether the microbial community of the sponge would be affected by the presence, absence, and acquisition and loss of the photosymbiont. To do this, we use 16S amplicon sequencing, and in the scheme on the top, we see the different phenotypes. Purple for cyanobacteria containing either originally or as a consequence of acquisition and transplant. White phenotype, original white sponges from the cave, or sponges that have lost due to transplant to the dark cave the photosymbionts, and the intermediate colonization condition. In the bars, we see in different colors the different phyla of bacteria found in these specimens. And what is evident is that the main difference are those green bars. Those green bars represent the cyanobacteria, and more specifically, Feldmanni, thus matching the phenotype of the sponge. However, at the phylum level, we don't see any remarkable difference in the acquisition and loss of cyanobacteria. When looking at the amplicon sequence variance level, we saw that photosymbionts, that the cyanobacteria did have some effect on the microbial community, yet they only significantly affected 2 amplicon sequence variants out of 1,000 approximately, and these 2 variants were at very low relative abundance. And therefore, we concluded that globally, the microbiome of the sponge was not particularly affected in acquisition and loss of the photosymbiont. So Spongarium, when lost by Chondrilla nucula, would cause the death of the host. Feldmanni, when lost by Petosia fisciformis, would not cause any damage to its host. And the microbiome was only minorly affected by the acquisition and loss of Feldmanni. So we wondered, what is the contribution of Feldmanni to its sponge host? So again, cyanobacteria photosynthesize, so that was the first direction. And in order to test whether the cyanobacteria produce carbon that is transferred to their host, we cut out of the sponge cylinders that would both contain the area with cyanobacteria as well as the internal area of the sponge that does not harbor the photosymbiont. We then incubated these cylinders in seawater containing C14 labeled bicarbonate, and then followed fixation of carbon in the outer and the inner layers. We did this for 2 different sponge species, both for Tionella swinhoy from the Red Sea that harbors Spongarium, and for Petosia that harbors Feldmanni. And we got very different pictures. In the case of Spongarium, there was fixation of carbon in the outer layer, but labeled fixed carbon could also be observed in the inner layers of the sponge, suggesting that Spongarium contributes organic carbon to its host. Differently, for Feldmanni, labeled fixed carbon was only found in the outermost layer where Feldmanni is found, and the internal layer were not obtaining any fixed carbon, suggesting that Feldmanni is not transferring organic carbon to its host. So what does Feldmanni contribute to the host? Well, according to the transplant that in highlight would not survive, we suggest that possibly Feldmanni provides a protection to the sponge from damaging light radiation. So to conclude, we've seen that Spongarium and Feldmanni have evolved from a common ancestor that has diverged to different types of both transmission modes, host localization, host specificity, as well as function within the holobiont, and that these different lifestyles can be seen expressed in their different genomic features. Furthermore, the facultative nature of Feldmanni enables the manipulation of the sponge-microbe interaction in a rather stable microbiome, opening a lot of new directions to study the interaction. And more specifically, what we plan on doing is to study symbiont acquisition, as well as the mechanism underpinning symbiosis breakdown, and also to look into the molecular regulation that underlies sponge-symbiont interaction, thanks to this manipulative model system. I would like to conclude thanking all the present and past lab members and highlighting in particularly in bold those that have specifically contributed to the stories discussed today, and furthermore all the collaborators with whom I have had and I still have the pleasure to interact on these and related topics. And thank you all for listening.
Thank you. Really exciting work on the acquisition of symbionts and also recognition by the host. So it'll be interesting to hear more of how this goes. So we will continue and jump now to Portugal, to Rodrigo Costa from at the University of Lisbon. And he's going to talk about distinct adaptive features of symbionts of marine sponges, specifically focusing on alpha proteobacteria. I see you have your slides up, and I think you are now in charge. Thank you. Unmute yourself.
Yeah, all right. Yeah, Max, thank you very much for the introduction, and thank you and FEMS for the invite to participate in this wonderful webinar. It is my great pleasure to take some minutes to share with you our studies on marine sponges, and in this particular case, alpha proteobacterial and symbionts of sponges. Let me just get a laser here. Okay, so these are examples of the sponges we very often work with. They all belong to the Dictyoceratida order, the so-called keratose marine sponges, Sarcotragus spinosalis, Actinia variabilis, for instance. So everything I'm going to talk about to you today pertains to those sponges or sponges in that order. So the core of this talk is obviously based on the study that we have published in 2018 in FEMS. I would like to start by thanking Elham Karimi, who was a PhD student in our group back in the day, working heavily on this data that I'm going to present to you today, and also Beate Slavi, Uta Henschel, who were major collaborators in this particular study. Moving on. Let me see. Okay, this is the one introductory slide that I will skip, obviously, because Russell gave us such a great presentation or introduction in sponges and in sponge microbiology and microbiomes. But I would rather like to make the point, though, that one of the key features that make us interested also in the marine sponge microbiome are their profusive biosynthesis or potential biosynthesis of natural products, such as polyketides, terpenes that may be synthesized by the microbial symbionts of marine sponges. So marine sponges and their symbionts are essentially chemically defended. The host is sessile, can't move, and because of that, there's a huge chemical diversity in sponges, and particularly within the microbial symbionts, which are now believed to be the main producers of natural products that may be involved in activities such as deterrence towards predation, inhibition of bacterial attachment, avoidance of fouling, overgrowth, and so on and so forth. Several bioactivities have already been reported for this wide range of natural products produced by bacterial symbionts of sponges. I leave discodermal lide here as an example of one polyketide showing potentially potent inhibition of growth of tumor cell lines, which is undergoing phase 1 trials. At this point in time, Dictyoceratida or keratose sponges, as I mentioned before, is one major focus of our studies from the host side. We are interested in this particular group of sponge species because they are very abundant and possess a high diversity in the Atlanto-Mediterranean region. That is where we are located. Several species. Back in the day when we started, there has been actually a paucity or in existence even of studies in the North Atlantic about these sponges, in the Mediterranean Sea as well. But luckily in the last 10 years or so, Patrick Irvin, Susana Lopes-Legentil, Lucia Pita, and colleagues, and also we on our side in Portugal, we have been studying a lot microbial diversity and patterns of microbiome composition in these sponges lately. And there's a lot of knowledge right now on microbial diversity on these sponges. Wide geographical distribution across this area, biogeography, spatial temporal patterns of microbiome, composition and distribution. They are very nice models to perform such studies. They are high microbial abundance sponges, and that is one key feature in the sponges that we like to study. I will show you in a minute why. And then in the past, already since the 70s, there's been a large inventoire of natural products with a range of bioactivities that have been isolated from these sponges whose origin remain unknown, right? Host, organism, or microbial symbiont. So what you see here is a typical skeleton. So one of the things that make the keratose sponges interesting is that their skeleton is made of a proteinaceous material, the spongin, filaments instead of spicules, and that is more or less the background that you see. I like to show this micrograph very often. What comes next are all the bacteria associated with that particular organism. The micrograph of Sarcotragus spinosulus, one of the model species we work with, and then look at the density of the alpha proteobacteria, right? The particular group we are talking about today, highly dense alpha proteobacteria and communities. In there, you will see also some highly diverse. Same for the gamma. So these are two of the largest, likely the largest groups of proteobacteria inhabiting sponges altogether, right? You see that these symbionts are hardly sitting on the spongin filaments themselves. They like to be in the mesohyal matrix of the sponges, and throughout giving you a background, right, to where we are at this point in time, right? So these questions 1 to 5, I consider them relatively well answered questions at this point in time. I mean, defining the alpha diversity of bacterial symbionts and these sponge species. We have been doing a lot of work with cultivation that I show you in a minute. A couple of trends we get from there, how much of this community can we cultivate, and whether these communities are species specific or host species specific or conserved. They are host species specific. Spatial temporal scales for these particular sponge species we study, so they tend to host their symbionts very well across biogeographical gradients and time. We have been doing a lot of comparative metagenomic studies to decipher the functional attributes of these microbiomes. This is also relatively well answered. I'm going to go through in a minute in a slide about it. But then the real question where we are at right now, so what are they doing, but who is doing what, right? The linkage between identity and function is what we are focusing on. And we have been putting some data around the alpha proteobacteria together lately, right? Yeah, so a little bit of a background. So what we have learned from amplicon sequencing microbial diversity surveys for these particular sponges, that is an example of one study of ours where we look into temporal dynamics and the capability of the host in maintaining its symbiotic consortium as a dynamic equilibrium, right? And the composition, as you can see here, so the keratose sponges are also very fun because they will contain at the phylum level all of those very fun taxonomic groups we like so much to see typically in a marine sponge, a huge bunch of actinobacteria, acidobacteria, phylotypes, the bacteria out there, a number of so far candidate phyla that we can detect in these sponges as well. And you can see roughly the composition of the community is relatively stable through consecutive years, also in terms of phylotype, richness, or diversity, what we can detect that it nearly doesn't change, so very stable symbiotic communities. And here are the proteobacteria and the portions that they occupy within this microbiome, the relative abundances altogether. So what we will see in this particular study we published in FEMS in 2018 is a little bit more particularly on the adaptive features of symbionts in the Hodospiralaceae family. to give you a glimpse or an idea, the alpha proteobacteria altogether make up from 8 to 99.5% of the total relative abundance in a given sponge microbiome. So these are assessments made on the basis of, based on the Nature Communications publication by Thomas and colleagues and Nicole Webster. So altogether, that's more or less the portion that alpha proteobacteria would represent. And within the Hodospirillacea, this particular family within the alpha proteobacteria may make up 11% of the total alpha proteobacterial composition in marine sponges, and worldwide we'll be dealing with a community of up to 420 different phylotypes belonging to this particular family that had been detected in this study published in 2016, associated with marine sponges, right? So a large diversity of symbionts belonging to that particular family. So there is a couple of things. So the study I will show you on the symbionts belonging to the Hodospirillacea family is based on this particular microbial metagenome sequencing. So like the total functional profiling has been published before. This is to give you a background, a typical comparative metagenomics study, contrasting the functional features of the microbiomes in Spongia officinalis sediments in seawater. And we knew from this profile that several very interesting functional features, some of them I have marked here with the red arrows, like the biosynthesis of polyketides, typical natural products with antitumoral activities, the biosynthesis of terpenes that may be involved in microbe competitive interactions. They are very or largely enriched in the marine sponge microbiomes compared with the environmental vicinities. We do have also indications for enrichment in genes involved in plasmid biosynthesis and also maintenance and stability. As Laura mentioned before, that is a typical feature of the marine sponge microbiome, like enrichment in CRISPR-Cas associated proteins. So we knew already from a general microbial community profile which typical features are enriched in this particular microbiome. The features we are highly interested in, such as those involved in natural product biosynthesis. A number of nutritional factors I can point out here also enriched in sponges and sediments in comparison with seawater. Aryl sulfatases involved in sulfur cycling, for instance. And the one remarkable feature in this particular study that came out was also in the total microbiome, right? The lack of the sponge microbiome being depleted in flagellar motility and chemotaxis associated genes, which for us served as an indication that this microbiome or the true mutualists associated with these sponges were, so to speak, as facile as their particular, as their hosts, right? So to answer the question who is doing what and focus on the alpha proteobacterial symbionts, so we have performed also now benefiting from Laura's study, right? We have performed the metagenome assembled genome-based study, which is the one published in FEMS in 2018, where we focus on this particular genomic being, right, assembled from those metagenomes belonging to the Hodospirillacea family, alpha proteobacterial family mixotrophic organisms, as I mentioned before, and we try to infer which what is special about this particular genome and other closely related symbionts in this family from sponges in comparison with their closest free-living relatives, right? So that is this study, and that is one phylogenomic tree that will tell you the clade which we are now studying and focusing on. All of these green entries here correspond to alpha proteobacteria beings assembled from marine sponges. In these groups here, from the Ateslavi Uterhenschel's group, this is our symbiont assembled from Spongia officinalis. They are all together here, making up one very robust, consistent clade where you don't have any culturable representatives and you don't have any seawater representatives in this particular clade. We're calling this clade the SERC, right? The sponge enriched Hodobacterialis clade, composed exclusively by symbionts sponges, unculturable microorganisms. And the point of this study was like picking from the databases out there the closest free living relatives available in seawater, and to compare and contrast the genomic features of these two genomic groups and understand which are the typical adaptive features of these symbionts. I should call your attention though to this number of other genomes in here marked in blue. So this is a group of a community of alpha proteobacteria, not in the Hodospirillacea clade, that we are able to isolate from these sponges, right? Using very specific, slightly different than usual cultivation methodology. So there's a huge diversity of also culturable alpha proteobacterial symbionts that we can isolate from these sponges, but not this particular clade. Cool. So just to cut a very long story short, when we pick like 3, that is an example of the ordination analysis of 3 symbiotic genomes, right? From this particular clade, you're picking this one, the closest relative, and that one. So 3 genomes from this clade, comparing genomic features by COG annotation, using COG annotation, genomic features of the symbionts against their closest free-living phylotypes. There is a number of specific, literally hundreds of clusters of orthologous genes, groups of proteins, right, so-called COGs, 700 specific COGs that we find in the free-living genome that we don't find here, 205 specific COGs or functions, if you will, that we will find in the symbiotic pool of genomes that we won't find here, and a number of them really showing statistically different quantities or relative abundances in these genomes. So, and just again, to make a long story short, some of the features which were remarkable and very clear, correspond to very clear trends to our opinion, was the complete absence, and here I'm talking about hundreds of genes involved in motility machinery, especially flagellar motility, that were absolutely lost in the symbiotic genomes, were present in the free-living organisms. And concerning, to pick one of the nutrient cycling, important nutrient cycles that we know that are very relevant for sponge metabolism and sponge microbiome dynamics, the capability of uptaking and breaking down or forming cleavage of taurine metabolism, which is a sulfated organic compound, was a specific feature and a highly significant feature in the sponge. Symbionts that we didn't observe in their free-living counterparts. Both these two groups of organisms, the Hodospirillacea, are very good at producing polyphosphate, accumulating polyphosphate. In both groups, we see this capability. So the Hodospirillacea may be also playing a role in polyphosphate formation within sponges. Several other important features involved in nutrient cycling, nutritional exchange between symbionts and sponges have been detected, such as sulfated polysaccharides, potential for assimilation of sulfated polysaccharides. A lot of genes are higher than sponge genomes being enriched for genes involved in antioxidative stress, degradation of aromatic hydrocarbons, and heavy metal resistance. So these were kind of top umbrella functions for which we have seen numbers and numbers of genes involved in these differences, and sponge symbionts kind of specialized or possess and enriched quantities of genes involved in these processes in comparison with the free-living organisms. So these are a couple of heat maps we have constructed and to show and display the one aspect that we liked so much to see in this study that has to do with nutrient metabolism, with the higher abundance of ABC transporters or transmembrane proteins involved in pumping in and out of molecules in the sponge symbionts in comparison with the free living, a higher diversity of such transmembrane proteins and a higher abundance as well, which to us served as an indicator that the symbionts are fine-tuning while adapting to the host, fine-tuning their nutrient metabolism through shaping their membrane composition and biogenesis is slightly different to be able to acquire and exchange nutrients in a more specific manner in association with its host. A repertoire of different channels that we can see in terms of abundance and diversity that change in comparison with the free living genome. So all of those channels are being enriched in quantities from nearly 0 to several copies of genes here involved in this particular transport systems, including branch chain amino acid systems, such as this one, leucine transport systems. And in terms of secondary metabolism, just to make a point in coming back to the issue of polyketides, we do also see evidence for enrichment of polyketide biosynthetic genes enriched in the pool of symbiotic genomes compared to free living organisms. We do see also here a higher counts or amounts of tau D, which is the gene involved in the cleavage of taurine. This sulfonated organic compound being enriched in the sponge symbionts that you see in orange here, in comparison with the free living organisms, and several other features involved also in the biosynthesis of antibiotics that end up being enriched in the sponge symbionts. So just to make the very last point of the talk and coming back to the issue of cultivation and this large panel of bacterial symbionts that we end up cultivating, alpha protobacterial symbionts that we end up cultivating from the sponges. That's another FEMS published article back in the day, 2013, revealing that when we do cultivate symbionts from the system, we do get and we do observe a lot of interesting bioactivities, antibacterial, antifungal, etc., you name it. So very nice consortium and isolates to study in the laboratory. But other studies from our team show us that only about 10%, and that's a very optimistic estimative, and we are optimistic about it. But anyway, only about 10% of the sponge symbionts can be readily cultivated in the laboratory. So there is a huge gap to be filled right there, right? And what is really dramatic in the case of the marine sponge microbiome is that exactly the most abundant symbionts, the very dominant ones, are the ones that we are unable to cultivate, right? We cultivate a huge diversity, extremely exciting group of microbes kept in the lab, but they usually belong to what we would call the rare biosphere within those particular microbiomes. Using specific strategies such as this one, I have just published very recently this study as well. So we dilute quite a lot the carbon content of the usual marine cultivation media, which are out there. We offer much less carbon, and we are able through these means to end up cultivating a large diversity of symbionts and large diversity of alpha protobacteria, right? Which do not belong, as you can see here. So these alpha protobacteria, huge area, under Sheniella, Erythrobacter, they're most often belonging to other clades, which are not the Holobionts family, but within the alpha protobacteria, which are also very versatile in terms of metabolism. But you can see here in our estimates of abundance in the marine sponge, like Sponge officinalis, none of these symbionts are truly abundant. Actually, they are kind of rare everywhere in the marine system, right? But it's slightly higher abundances in seawater than in the sponges, but they are there, and they may be transient symbionts popping up once in a while, not really highly dominant. Interestingly, we can cultivate them and study their genomic features as well, right? In terms of secondary metabolism, this alpha protobacteria, we are dividing here in 3 different phylogenetic groups according to their families, and so on and so forth. Here are the Holobacteriaceae, second group, third group here the Sphingomyonadales, and so on and so forth. inexorably, all of them are very interesting in terms of secondary metabolism and potentially capable of synthesizing natural products that we know are enriched in the marine sponge microbiome, right? And not only this, not only secondary metabolism, but also several other traits that we know are remarkable and hallmark features of the marine sponge microbiome can be readily identifiable in this group of low abundant culturable alpha proteobacteria. These features we have been seeing in our Hodospirillacea symbionts or in the total microbiome as enriched in the sponge microbiome is present in all of these alpha proteobacterial groups, right? Potential for antibiotic biosynthesis, yes, sulfur metabolism and cycling, taurine deoxygenase, taurine acquisition and cleavage, and so on, resistance to antibiotic, etc., metabolism of aromatic compounds, all of those features which are typical of the most dominant sponge symbionts are also present in this low abundant, readily culturable alpha proteobacteria, which makes us think they may be a resource of functional redundancy within the marine sponge microbiome, right? So the one thing that they can do, rather, like all of these features involved in motility and chemotaxis are there, right? They are typically planktonic. They may be living across these three different phases, sediments, seawater, and sponges. They will possess this dual lifestyle, as Laura also mentioned, right? Possessing both genomic features for competent free living or association with marine sponges, right? So this rare biosphere that we can cultivate from the sponges may be a reservoir of functional redundancy within the system. So to conclude very briefly, so metagenomic binding, and comparative genomics of the symbiotic alpha proteobacteria, they're all highly congruent in that we find polyketide, terpenoid, biosynthesis of remarkable features of this group, lack of motility and chemotaxis, especially for the true mutualistic symbionts, and important participation in sulfur cycling, right? So in the future, we will explore this nutrient metabolism of the alpha proteobacteria to try to capture the true symbionts in the laboratory. We know that this is a very hard task, but we are heading towards this goal. And I would like to thank all of my collaborators that have been involved in this work throughout the years. And I would like to thank again all the panelists and Max for the opportunity, and everyone for your attention. Thank you so much.
Well, thank you. This is really a nice selection of different aspects of sponge biology or sponge microbiology. So as we go in, you can send the questions through the question link, and I will start to go through them and start to sort out, and we can have this discussion on there. Just while we're beginning and I figure out how to navigate the question panel, Russell, you mentioned aerobic, anaerobic conditions within the sponge animal. And I'm wondering how important is the redox cycling that likely occurs with pumping or non-pumping conditions in the sponge for all of these nutrient cycles?
So Max, I suppose I shouldn't be surprised with you asking about redox, but I believe that it's likely very important, and I think that the sponges have a whole lot of control over that. And this is something we didn't mention. Sponges have exquisite control over their pumping rates, and are very actively changing, I think, the redox potential and the amount of aerobic and anaerobic material within the sponges. But that's something that some other groups have looked at, and it's something that we really haven't studied at all. Maybe Laura or Rodrigo would have something to add on that.
One thing that I know, not from our studies, but from studies of Gitai Yahel, is that in addition to the control of the sponge on the aerobic and anaerobic conditions, The group of Gitael has found that the sponge can go anaerobic even while the sponge is pumping seawater. So that raises a question whether, in addition to the control that the sponge has, that the microbiome might also create an anaerobic condition while the sponge is still pumping seawater. But I think that's a whole novel area of research. And
yeah, well, I'm thinking of even a wastewater treatment plant, an activated sludge system that's continuously aerated. There's virtually no dissolved oxygen in the system. So there's so much organic carbon that it's just being sucked up so quickly. And that's why also there are so many anaerobes. There's a question on adding to the nutrient cycle. So you talked about nitrogen and phosphorus. There's a question, what about the sulfur cycle?
So why don't I start with that? We do cover that a little bit in the review that we published in FEMS Microbiology Ecology that underlies this webinar. It's not something my own lab has worked on, and I can tell you, I think it's a really understudied. So at this point, we know a lot about carbon cycle and nitrogen cycle, but phosphorus cycling and sulfur cycling are studied far less. There is some good work on that. Some of that's been done in Israel and by other European groups, but I think it's an area that really warrants some additional study.
Okay, and then to actually follow up also about nitrogen fixation. So different than the plant symbiotic, the ecosystems, of course, are significantly different. So how would you compare in terms of the associated nitrogen fixers in a plant root or phyllosphere system versus the sponge?
So I think I would say that, as with so many things, sponges are way more complex. And in most cases with plants, they have very highly evolved and efficient nitrogen fixation systems, but often driven by single microbes. You know, Frankia come to mind, where you have very specialized nodules that are extremely efficient in nitrogen fixation in some leguminous plants. But in the case of sponges, we have these incredibly complex communities with several hundred different bacterial symbionts in a single sponge. And as I mentioned briefly in passing, we see big changes in patterns of NIF-H gene expression during the day, at dawn, during the middle of the day when there's a lot of light. And I think the patterns are very dynamic, and there's an interplay between many different bacterial symbionts. So I'd say the sponge system is more complicated and more interesting, but perhaps as a result, a little bit more difficult to pull apart. Just finally, I think it is becoming very clear that the cyanobacteria are absolutely key in the nitrogen fixation processes in many different sponges.
Okay, and here's actually a comment from somebody we all know, Ute. She's putting you all on the spot here. So why can sponge symbionts not be cultivated? Genomics reveals no evidence of auxotrophies or genome reductions as far as is known. And maybe we don't know the answer. So this is to all of you. What about the cultivation or unculturable organisms, or not cultivated yet?
I'll start with a sentence or two. I think both Laura and Rodrigo will have a lot to say on this, but basically there are not enough people trying. People have given up on that, and it's so easy to get amazing insights from the molecular approaches, but there are huge advantages if you are able to actually culture these symbionts. It's a lot of effort, far more difficult than much of the molecular work at this point, but I'm very interested to hear what Rodrigo or Laura have to add to that.
Yeah, you want to go first, Laura?
You can go first. It's okay.
Yeah. All right. Yeah, my opinion on that, as you have seen, we have been cultivating a tiny bit throughout the years. It's not something we are going to give up on because, as mentioned, even though, I mean, my perspective as well, I mean, in this lifetime, who knows, we are going to be able to cultivate those through highly abundant symbionts. Again, to emphasize it is really dramatic. I mean, we have studies where we are just like sequencing 6.5 amplicon sequences of the very alicoats that we put on the plates that we use to plate. On those alicoats, we know that all of those symbionts are there. We try to cultivate and they just don't grow. We end up, that this trend is very interesting. We tend up to cultivate what is the rare biosphere of the marine sponges, some phylotypes that we wouldn't be able even to detect using amplicon sequencing because they are in much lower abundance than that. They are all very interesting, including this high diversity of hair proteobacteria or alpha proteobacteria. So we are actually, at this point in time, using cultivation more as a means of sampling the rare biosphere within the marine sponge microbiome rather than really wanting to pick the highly abundant ones. So that's the approach more or less at this point in time, because as Russell said, it is so difficult. I mean, the usual steps now would be, now that we know so much about the microbial metagenomes and we learn more about nutrition, right? And picking on the sulfur cycle, for instance, those evidences that the microbial symbionts may be processing sulfated polysaccharides, maybe importing and cleaving taurine, right? And so on and so forth. Start creating culture media that will use those nutrients in that composition in order to try to cultivate them better. But then you will run into a second problem, which is the redox problem, right? And how complex that is. So far, for instance, in our team, and that's why it is so difficult. In our team, all of our cultivation attempts so far have been done in complete aerobiosis, right? So to implement one culture collection and take care of them and curate them is a lot of work, years of work, right? I start a new cultivation attempt using microaerophilic conditions, for instance, which I think is something that is needed, right? But then you know you're going to go through those many years characterizing that particular consortium. But these are the little bits that need to be added to the puzzle. And I guess it is only through the concerted action of several different groups doing different things that will enhance our ability to catch those very dominant symbionts that we want so much to have in the lab, right?
I can add only something small. So there can be a lot of complementarity in the metabolism of the symbionts that live together in the sponge. And therefore, there can be so many little things that we would need to guess that a symbiont is requiring and that is provided by a different symbiont. So that's one issue. The other issue is that even when we use the genomic information, a lot of the genes are not necessarily annotated, and we might not be able to decipher what is really missing. We have tried also for a long time. We keep not giving up. Specifically for the Synechococcus symbionts, we have tried to adapt and utilize the genomic information that told us some of the differences, and still haven't achieved the cultivation. So it is a big challenge. Another thing that I'm wondering about is the high amount of chemistry found in the sponge. And there might be a way in which the symbionts inside the sponge are protected from this chemistry. And I wonder whether the moment we take apart and homogenize the sponge to isolate the symbionts, we are not quickly putting them to face this chemistry, and they might not survive that very first step before we actually transfer them to the culture medium, which could be an additional obstacle.
There are two essentially follow-up questions then here. One, is it possible that the secondary metabolites produced of bacterial origin in sponges are a result of change in physiological conditions, or a way of microbes prevented from self against some change? And could this be the reason those of bacterial origin of biomolecules cannot be cultured? So maybe it's to Rodrigo in terms of, yeah, is it that they're making key molecules, really their bacterial origin, and then you're taking out the other bacterial partners in a sense. That's how I interpreted this.
So, and if that would be a factor preventing them to grow?
Of cultivation.
Well, I wouldn't think, if I understood the question correctly, that the fact that one particular organism or symbiont in this case, because it is eventually producing a natural product that would prevent that organism to be cultivated, you may think that maybe competitive interactions in the culture medium, right? But we do also isolate plenty of organisms producing polyketides or producing bacteriocins, terpenes on culture medium. Likewise, I guess there's perhaps not the natural product biosynthesis per se, but more intricate features or factors that derive from the host microbe interaction, as Laura mentioned, some factors that may enable the symbionts to establish itself and adapt and live very well within the sponge. And those features that we completely don't know what they are get lost, or also secondly, specific nutritional features that we are missing, and also aerophilic, microaerophilic, anaerobic conditions that may favor the growth of particular symbionts under these circumstances. So yeah, that's my take on it, if I understood the question correctly.
So there's another one Lee Kirchhoff had asked on the sponge microbiome appearing to be very active and presumably growing quickly. Yet the overall microbial community is stable over time and space. Can the panel comment on whether it's the host or the endosymbionts which are largely controlling the composition of the community?
I was going to comment on the, if I understood correctly, on the fact that the symbionts would be growing fast in the sponge. And I don't think that there is any evidence of the symbionts being growing fast. And if we look at the transmission microscopy images that also Russell was showing and that are found in the literature, you hardly see any dividing symbionts. So I would rather think that those symbionts are oligotrophs that divide and grow rather slowly inside the host. Not that I have direct evidence, and there might be actually today some metagenomics type of analysis that might enable to determine cell division rate actually in uncultured symbionts. But as far as I know, nobody really has utilized them in the sponge microbiome.
Yeah.
Regarding who has control, I don't really think that we know who has control on symbiont division.
Well, if I can share 2 words about that, I guess, well, in terms of control, I think that is a two-way street. Both host and symbionts are, have a major role in the process. So there's been studies also by Thorsten Thomas and groups, recent studies by Ute as well, showing very well like the presence of some genetic elements such as eukarine repeats, right, that allow the symbionts to avoid phagocytosis by the sponge host. For instance, that is one of the major and true mechanisms known so far that allow symbionts to establish themselves within the marine sponge microbiome in the host. That is kind of the symbiont is actually evading phagocytosis by the host, so you can count that as one of the ways in which the symbionts are taking part in this particular relationship. As far as the microbial community is growing within the sponges, I guess Russell may be in a better position to answer that. My belief, I sense that the microbiomes are usually very dense, especially if you're talking about high microbial abundance sponges, right? But the symbionts tend to stick there somehow. Not much by division, as Laura mentioned, but I do believe they are highly active, right? And we are talking about here of a metabolism that is very fast eventually, highly active, but that metabolism is not being invested in population growth, but more in metabolic processes. But there may be a paucity of studies dedicated to that, but that is my guess of what metabolically speaking might be going on for the majority of the abundant symbionts in the sponges.
So I think it's a very interesting question, and I think Laura and Rodrigo have covered most of what we know. There's a lot that's not known. I want to add just one thing, and that's we should never forget the phages. It's becoming more and more clear what an important role bacteriophages have in the sponge microbial community, and they could be part of the dynamic that determines the rate of turnover of the symbionts.
So as that note, bringing up viruses, I do think that we need to wrap this webinar up. It's been wonderful. Thank you, Laura, Rodrigo, and Russell, for your insights and time. And thank you, everyone who's attended this webinar. There are a few more questions that were not answered, so we're going to try to sort through them and we can get back to you. And of course, also, please feel free to contact any of us for other questions that might still be left. I just want to alert you to our next series for something completely different, Polar and Alpine Microbiology on July 23rd, and then Environmental Dimension of Antibiotic Resistance on August 6th. So take a look at the FEMSmicrobiology.org website for links and so on that. And with that, thank you very much, and stay safe, stay healthy, and hope to see you all in person on a sponge conference sooner rather than later.
Thank you so much.
Thank you very much.
Thank you everyone in the audience. Thank you. Thanks, Max.